Welcome, everyone, and thank you very much for joining our UC Health System-wide Grand Rounds. Our topic today is avian flu, an emerging and evolving threat to humans and animals. I'm Deena McRae, the Associate Vice President for Academic Health Sciences at the University of California Office of the President. UC Health hosts a series to help keep our community members and colleagues up to date on hot topics and issues that may impact our professions and communities that we serve. This Grand Round series also allows us to showcase some of the extraordinary, innovative work that is constantly being performed across our UC system, work that aspiring other educators, researchers, and clinicians across the nation and around the world, as well as improving care. I want to thank everyone who helps to create and support these quarterly UC system-wide Grand Rounds. This is a CME in Veterinary Medicine continuing education event. After this session, so you can provide feedback and get continuing education units for participation, you will receive an automated email tomorrow to claim your continuing education units. Time will be allotted for answering questions at the end. Please feel free to use the Q&A feature for entering questions throughout the webinar, and we will answer as many questions as possible at the end of the presentations. The speakers do not have any disclosures. I am honored to be here today to showcase 4 expert leaders from our UC Davis School of Veterinary Medicine and School of Medicine, here to discuss avian flu, the impact on our communities, and what UC is doing in response. First, we'll hear from Dean Stetter. Dr. Mark Stetter has served as the Dean of the UC Davis School of Veterinary Medicine since 2021. Previously, Dr. Stetter served as Dean and Professor at the College of Veterinary Medicine and Biomedical Sciences at Colorado State University. He also served as Director of Animal Operations, Director of Animal Health, and Clinical Veterinarian with Disney's Animal Program at Disney World. A diplomat of the American College of Zoological Medicine, Dr. Stetter's research interests have included advancing minimally invasive surgery in non-domestic species and a variety of animals, including fish, reptiles, birds, and mammals. His work has resulted in more than 100 scientific publications and conference presentations. We will then hear from Dr. Ashley Hill. Dr. Hill is the director of the California Animal Health and Food Safety Laboratory at UC Davis, where she is also the Associate Dean and Professor of Clinical Diagnostic Epidemiology. Her research interests include equine musculoskeletal injuries and diagnostic testing for livestock and poultry diseases. She will be followed by Dr. Christine Johnson. Dr. Johnson is a professor of epidemiology and ecosystem health at UC Davis and focuses on the intersection of emerging disease and environmental health. And finally, we'll hear from Dr. Angel Desai. Dr. Desai is an associate professor in the Division of Infectious Diseases, associate hospital epidemiologist, and director of global health for medical students at the University of California, Davis Medical Center. We are honored to have these impactful leaders with us today. I will now turn this over to Dean Stetter.
Well, good afternoon, everyone, and thanks, Dr. McRae. Hey, let me start with a couple of big picture items as it relates to avian flu. First of all, avian influenza is not new. It has been around for decades and has been circulating in wild birds, especially birds that migrate across the globe. But what is new is this virus is changing, and this changing in multiple events that have raised concerns lately. So we have seen, for example, how it has jumped from avian species now to cows, which was not on our radar, cats as a domestic pet in our homes, seals, and other species that we'll talk about. It's also begun to jump to humans. And while getting conjunctivitis from a milk sample that has a high concentration of virus may not be surprising, we are seeing a variety of cases that we don't necessarily know the source, and there's more and more concern about how it might be exposing humans in ways that we hadn't predicted before. The other concerning area that we'll talk a little bit about is direct transmission. We have seen in wild populations, specifically in seals, that while it might have started by consuming a wild bird that had avian influenza, but there's more and more evidence that there has been direct transmission between seal species and mammals, causing mass mortality effects. Now, as a professor of zoo and wildlife medicine, I'm often thinking about comparative medicine and the differences between species in categorizing infectious disease as it relates to zoonotic things. We know that zoonosis is common. We know that most of them are from bacteria, and that when we think about viruses, it is pretty uncommon for a virus that has zoonotic potential to go from what we would consider the lower vertebrates, from fish and from reptiles and from birds, into mammal species. So that is another area of concern when we think about spillover or changes that we've seen. So for the purpose of discussions for today, we're going to talk about a variety of different animal groups. I'm going to lump them into four. We'll talk a little bit about the pets. We'll talk a little bit about food and animals that are part of our food systems and agriculture. Talk a little bit about wildlife and how avian flu is affecting free-ranging wildlife across the world. And the last group of animals would be our human primates. And obviously we have public health concerns around this. Now, how this virus affects these different groups is tremendously different. And we'll learn, for example, that a cat who drinks a contaminated milk sample with avian influenza in it often becomes neurologic and typically is a fatal disease. But many other animal species, for example, our domestic dog, have not been proven to be infected with that. So let's jump right into some additional details. Next slide, please. So looking at the image in front of you here, these are our three big categories across the US on what we've seen with avian influenza. So for several years, we've seen poultry cases continue to become a major issue across the US. So when you look at these maps, you'll see that anything with color shows that there's been positive cases of H5N1 in those states, and the darker, the deeper the color, the more cases there have been. If we go to the middle, really the dairy cases just started almost exactly a year ago, and that was a surprise. For dairies, this was not on our radar. These are not animals that from a surveillance perspective we had tested for avian influenza before. And these are animals that didn't get terribly ill from the disease, but had a transitory couple weeks of reduced milk consumption and flu-like signs compared to poultry that become very ill and often fatal. And then just in the last 6 months, what we've seen with the evolution into human cases. And what I want to point out in all three of these maps is California, and the fact that California has had a high incidence with all of these different groups, and that just here in the last couple months, the governor has declared a state of emergency as it relates to H5N1 and the concerns as it relates to animal and human health. Next slide. So why should we care about this, especially if at the moment is not deeply affecting human health? Well, when we think about California, we literally feed the nation, whether that be through poultry meats, eggs, dairy products. We are the leading producer of food in those areas for the country, if not many areas around the world. So how is this affecting our food safety security? What are the concerns as it relates to animal health, and then the impacts to human health, and also the economic pieces here? Just so you know, your UC Davis School of Veterinary Medicine is the only public veterinary school in the state. We have, and we'll talk a little bit about this with Dr. Hill's presentation, but we have veterinarians throughout the state that are working in multiple areas to make sure, whether that be diagnostics, whether that be disease modeling, whether that be biosecurity, vaccine development, that we're helping keep our food safety, our food safe and secure. Next slide. So if we drill down to California specifically and talk about the impact it's had, just in the last couple of years, we've had to euthanize over 20 million birds. And if you've been to the grocery store lately, you know that there are many shelves that are empty as it relates to eggs. So the age-old question about what comes first, the chicken or the egg, I cannot answer, but I can tell you that there are, after euthanizing millions and millions of birds, that the egg prices and the egg availability is being directly impacted across the state. And if you look at the dairies, what's really remarkable is that has just been happening really over the last year. And so here in California, even less so as far as time-wise, and it has really exploded as far as the number of dairies and the number of animals that have been affected and the economic impact. And then we'll talk a little bit later about how that obviously is also affecting the number of people that have become affected. I have given a presentation on this several times over the last couple months, and each time I give this presentation, that number right now, 67, is also out of date from when the slide was created, and those numbers continue to increase. Next slide. So I want to focus your attention on the bottom right where it says species. What I want you to take away from this busy slide is this is the mammal species that have been affected by avian influenza. And this includes both captive pet species, for example, you'll see domestic cats here, and also free-ranging wildlife. So black dots are a good example of the number of cats that have become neurologic, that have died from this disease, and typically that has been from consuming unpasteurized milk. At the same time, we've seen a variety of other wild species. You can see raccoons, you can see skunks, you can see other animals here that probably ate an infected bird and became infected. So this is not just with birds and not just with a specific species of mammals, but it is affecting multiple animals all over the place. Next slide. So I'm going to take you down to South America for a moment, and we're going to talk a little bit about how this is affecting free-ranging wildlife. We have a wildlife veterinarian in the School of Veterinary Medicine who is stationed in South America and who noticed 2 years ago mass mortality events with marine animals along the coast of Argentina, Chile, Peru, and in further diagnostics, what she was able to determine was that while it started with wild birds, here in this picture you can see terns that were finding in large numbers along the coastline. It then moved to marine mammals. And then what we saw in seals was literally thousands and thousands of animals that were dead or dying that were washing up on the shores of Patagonia and in South America. And she's published in the last year or so her findings here, which show, number one, that over 97% of the neonates of those seals passed away, and that there is strong evidence for transmission between the animals, whether that be between adults or being passed in the milk and the neonates being affected. So while this has been a major event, 17,000 animals died along the coast there, we have not seen the same level with marine mammals here in California. And you can imagine that if we had 17,000 seals that were floating up along the coast here, we would have heard about it in the news much earlier. Next slide. So one of the things that I really love about today's rounds is the fact that we have almost 2,000 people who have signed up for this event. And that includes physicians, it includes scientists, it includes veterinarians, it includes public and media, it includes other health professionals. And it's a great example of how complicated diseases like avian influenza are affecting our world, but that need a multi-pronged approach and that need health professionals from across multiple disciplines to come together to understand what happens with migratory birds in human health and agriculture in our pets. So our One Health Institute is a great example of a place here at Davis that helps combine that multidisciplinary approach, and you'll be hearing from our faculty today that are part of the One Health Institute. So I'll turn it over to Dr. Hill, and again, thanks to everybody for your interest and for joining us today.
Thank you, Dr. Stetter. As Dr. McRae mentioned, I'm Dr. Ashley Hill from the School of Veterinary Medicine. I'd like to start with an overview of our veterinary diagnostic laboratory and the role that we play in outbreak response before I move into the specifics of H5N1 influenza. We are a system of 4 laboratories located strategically throughout California to be near the heart of California's animal agriculture. We're operated by UC Davis and subsidized by the California Department of Food and Agriculture. Our mission is to protect the health and productivity of California's livestock and poultry, safeguard public health, and diagnose animal diseases. Big picture, we have 2 big jobs. We are essentially CSI for livestock and poultry, receiving carcasses and samples and determining the cause of death or illness for our livestock and poultry producers. We also function as the smoke alarm for the California Department of Food and Agriculture's fire truck. And by that, I mean that we are constantly looking for foreign and emerging diseases in the samples that we receive. When we find something unusual or concerning, we alert the California Department of Food and Ag, and they take regulatory action for outbreak control. We are a part of 3 nationwide networks that serve the public good. The National Animal Health Laboratory Network, under USDA, is a partnership of over 60 public animal health laboratories that's capable of testing large numbers of samples for specific foreign or emerging disease agents originating from food animals. As part of the Food Emergency Response Network, under the FDA, we respond to biological and chemical contamination of food. And last, as part of the Veterinary Laboratory Investigation and Response Network under FDA, we respond to animal illnesses caused by foods or drugs. Here's a typical work year for us. More than 11,000 necropsies, which are animal autopsies, across a range of species. More than 450 foreign animal disease investigations, more than 350,000 tests across a range of diagnostic disciplines, 8 outbreaks, and 2 food safety detections in dairy products. And right now, we're doing a lot of diagnostic testing for influenza A in poultry and dairy on top of our routine diagnostic work. Okay, so moving on to H5N1 influenza in farm animals. What is going on? First, as Dean Stetter mentioned, I think it's important to recognize that the current H5N1 outbreak has been going on for longer than you may realize. This virus has been circulating globally since 2020 in migratory waterfowl, such as geese and ducks, and it's been spilling over into other wild birds, into a range of mammals, and into domestic poultry for years. It's making the news now in large part because we're seeing sustained mammal-to-mammal transmission in cows, and that's something new. So I want to start by comparing the current H5N1 outbreaks in commercial poultry versus in dairy cows. The current H5N1 outbreak in poultry is global and has been ongoing in the US since 2022. In contrast, the dairy outbreak started in early 2024 and is only affecting the US. Other countries have not been affected. The poultry outbreak consists of repeated spillovers from migratory waterfowl into domestic birds with minimal flock-to-flock spread. In contrast, the dairy outbreak is almost entirely the result of one spillover event from wild birds into a single dairy in Texas, and then rampant dairy-to-dairy spread and frequent spread from dairies back into domestic poultry. We've had 2 additional spillovers from wild birds into dairy cows in the last couple of weeks, one in Nevada and one in Arizona. In chickens and turkeys, this virus causes high mortality, up to 90%, and rapid onset of clinical signs. The rapid onset means that the disease is detected quickly. In dairy cows, the onset of clinical signs is slow, and the mortality is much lower. The slow onset means that without active surveillance, disease may not be detected right away, which allows it to spread. In birds, the H5N1 virus is shed in feces, saliva, and nasal secretions, whereas in cows, it's shed in milk. There may also be other shedding routes in cows. This is being investigated. Because of differences in mortality, detection, and spread, there are slight differences in the disease control measures for H5N1 in poultry versus cattle. For both species, control measures for an affected farm include contact tracing, biosecurity, and regional movement controls and testing. For poultry, an affected farm is depopulated. The birds will die of illness otherwise, plus the depopulation can prevent amplification of the virus because disease is typically caught early before the majority of animals in the flock are infected and shedding. For dairy cows, the farm is quarantined until repeatedly negative for the virus. Most cows will recover from their illness, and on both poultry and dairy farms, occasional human illness is detected, primarily in farm workers. So here is where the virus has been detected in the past month. These maps are slightly different from Dean Stetter's. I think Dean Stetter's covered a larger time period, but I'm just going to look at the last 30 days. So the map on the left shows dairy detections with darker blue indicating more detections. You can see that California is clearly number one, with more than 30 dairies newly affected. Remember, in cows, it's mostly spread dairy to dairy, and we have a lot of dairies in California with a lot of contact between them. The map on the right shows poultry detections in the last 30 days, and you can see that's much more dispersed, reflecting repeated spillovers from wild birds to domestic poultry. So I wanted to talk about how surveillance testing of dairy and poultry is currently functioning in California. When veterinarians detect sick birds or cows, they submit carcasses and samples to our California Animal Health and Food Safety Lab. Any positive H5N1 detections are reported to the California Department of Food and Agriculture. When a farm is determined to be infected, a control area and a surveillance area are marked on a map, and additional testing and movement controls are applied to other farms in those areas. Poultry premises in a control area are tested weekly. If any are positive, they become centers of their own control and surveillance areas. On the dairy side, all raw milk dairies in California are tested weekly, as are all uninfected dairies within a control area. California is working to have all dairies in the state tested weekly. Once a dairy is infected, it is released from quarantine after testing negative on 3 consecutive weekly samples, and after being released from quarantine, it's tested every other week. Nationally, the U.S. is implementing a national milk testing strategy. This consists of bulk milk testing nationwide to determine whether states are affected or not, followed by state-specific surveillance plans for affected states. So I know one topic that comes up a lot is whether we need to be concerned about H5N1 in the food chain. And the short answer is no. On the poultry side, cooking inactivates the virus, and sick birds do not enter the food chain because infected premises are depopulated. Any poultry premises in control areas undergo lots of pre-movement testing before birds or eggs go from the farm to market. On the dairy side, pasteurization inactivates the virus. Milk from sick cows does not enter the food chain, and that's for any illness. Any sick cow needs to be segregated so that its milk does not enter the food chain. On standard dairies, milk is pasteurized, and on raw milk dairies, samples are collected weekly for surveillance testing, and if they're positive, the dairy is not allowed to sell raw milk. However, as Dean Stetter mentioned, H5N1 is having an impact on the food chain via the price of eggs. Lots of poultry farms have been affected. Poultry farms housing more than 22 million laying hens have been affected in the last month. With each hen laying an egg a day, that's more than 150 million eggs a week that disappeared from supermarket shelves in the last month. So I wanted to talk briefly about what is being tested and how. On the poultry side, oropharyngeal and/or cloacal swabs are tested. 30 birds per house are sampled. And on the dairy side, the samples are milk samples, and those are either coming from the bulk tank, the silo, or the creamery and processor. On the right, you can see a graphic of a milking cow and then how that milk is collected into a bulk tank. It's then put on a truck and then it goes to the processor. We're testing at various stages along that chain. The test itself is an influenza A matrix PCR, and if that is non-negative, we do subtyping PCRs for H5 and H7. We also do a strain-specific PCR for H5N1 for the current outbreak strain. Anything that's non-negative is sent for confirmation to the National Reference Lab, which is located in Iowa. Sorry. And then both the National Reference Lab and our lab are doing sequencing to look at what's going on with the virus and to help identify when we get new infections, whether the source of the infection is wild birds or from infected cattle or poultry. The cattle clade has been fairly consistent, and it's pretty distinct from the poultry wild bird clade, which has continued to reassort and change and evolve. The livestock and poultry testing is being done in our lab as well as other labs within the National Animal Health Laboratory Network. So what can you do to stay safe? Basic food hygiene is great, and it doesn't just protect you from H5N1. It's great for all kinds of foodborne diseases. So cook your meat, keep your hands, utensils, and surfaces clean. Wash your hands after handling raw meat and separate. Don't chop your salad veggies on the same cutting board that you use for raw meat. I would say that raw milk does carry risks. We do weekly testing, but with samples a week apart, it's still possible that milk with the virus can get into the food chain. And cats are specifically very susceptible to it. The virus remains viable in raw milk. I would recommend that you don't feed raw milk or a raw diet to cats in your household. And I'd also recommend avoiding exposure to sick or dead animals. If you need to be near sick or dead animals, please wear appropriate PPE. That includes sick wild birds, poultry, other birds, livestock, and cats. Appropriate PPE would include an N95 respirator and safety goggles. If you are exposed to sick or dead animals, monitor yourself for conjunctivitis and/or a new respiratory illness. Here are some actions you can take. Here are some numbers, 2 numbers that you can call if you see sick or dead animals. The first number is specifically to report sick or dead birds, and the second number is for all wildlife. Call your vet if you think your cat is sick and may have been exposed to H5N1. There's lots of good information on this virus available publicly from many agencies and entities, including the CDC, USDA, California Department of Food and Ag, California Department of Public Health, and ProMedMail. And then last, who is at the highest risk of exposure? That's people who are likely to come into contact with sick or dead animals or products from those animals. That includes poultry and dairy farmers and workers, veterinary practitioners and their staff, regulatory veterinarians and animal health responders, and workers handling raw milk or other contaminated materials, such as food processors, slaughterhouse workers, and dairy laboratory workers. I'd like to welcome my colleague, Dr. Johnson, who will focus on the impact of avian flu on our wildlife populations.
Thank you, Dr. Hill. I was hoping to share just a little bit more about how much has changed across the globe over the past few years, as Dr. Dean Stetter mentioned, that all of this has facilitated the situation just highlighted by Dr. Hill, which I think can help us a little bit understand what could come next. So this shows the usual scenario for avian influenza, which is really well highlighted here, I think, by this figure by the World Organization for Animal Health. As Dr. Hill noted, there's routine transmission of low-path viruses, which have naturally evolved for centuries in wild birds, meaning that they're lowly pathogenic, so we call them LPAI, low path. And we have occasional spillover into poultry farms where they have on occasion evolved into these high path viruses in intensive production facilities. The epicenter for this typically has been Asia in the past, and this back and forth transmission between wild birds and poultry occurs in many parts of the world, as in large scale production facilities, as well as in more rural farming situations. So I just show here in these pictures some of the photos taken from a community that I worked with who were living in temporary settlements on a river in Nepal. And just for example, their livestock, including ducks, range free, mingling with their wild neighbors during the day on the river. And then at night, they're kept in this closer contact in these pens overnight. And so this back and forth transmission has enabled this new emergence and development of new clades over time. Next slide, please. So while the usual scenario was cause for concern, and even more worrisome scenarios started to emerge in 2021, as was highlighted by Dean Stetter. So I'm showing just a few of the notable events indicating that something unusual was afoot. So when the clade H5N1 called 2.3.4.4b initially arose out of Asia many years ago, there was started to see a surge in outbreak activity outside of poultry farms, including starting in 2022 in seabirds near the Caspian Sea. There was a huge die-off of terns and gulls, and then shortly thereafter throughout Europe. And this version was then becoming increasingly detected in many different mammalian and bird species, including in a mink farm in Spain, which was published with suspected evidence for mink to mink transmission, which was also really unusual. The first indication that we were then entering an entirely new scenario for H5N1 was when sea lions and pelicans began dying in extraordinary numbers in Peru, which then shortly progressed into many major mortality events across South America, coastally and marine species. And then upending any idea that we had of business as usual for this virus. As we've said, infection was detected in dairy cattle about a year ago, initially in Texas. So this map, I'm just showing the reported cases in wild birds over the past 2 years. And while the numbers are somewhat impressive, this is of course just a tiny fraction of the actual number of cases that would have occurred on the ground. Next slide, please. And we just, I think, all want to highlight the marine mammal outbreaks were really the first harbinger that the epidemiology of H5N1 in mammals was dramatically changing. First off, this virus had never made it as far south in the Americas. And second, this level of marine mammal outbreak was unprecedented. And this is a word we see all too often these days, but I'm hoping it does not lose its meaning, especially in this context. And as we said, all of this work was led by Dr. Marcy Uhart, has been working to help countries around the globe prepare for and respond to animal influenza outbreaks in her role at the World Organization for Animal Health. She also personally responded to marine wildlife events in Latin America, helping countries deal with this extremely, extremely challenging virus. And her efforts were able to track the virus all the way around the tip of South America. And of course, increasingly concerning news coming from infections in Antarctica with very vulnerable species that are there. Next slide, please. So unfortunately, we are now in a new, much more complicated scenario for H5N1, where we have sustained high path transmission of the virus. So now a high path version that has spread across not just wild birds, but spill over into seals, sea lions, and many domesticated species, including cows, which has led to this sustained transmission chains, which from an epidemiologic standpoint are the most concerning thing. Mainly because this can enable these new evolutionary pathways, which make it even more difficult to monitor what's happening, discern trends on the ground. And there's every indication, unfortunately, that this new scenario will only get more complicated with time. So rising with cases in cows or cats, which I think was covered really well, but just a helpful reminder of just how fundamentally helpful pasteurizing milk and cooking food is in keeping both us and our pets safe. So next slide, please. I just wanted to place H5N1 into the context that I always place emerging infectious diseases in, and these all sort of tend to follow this same sort of pattern, including ones that go on very rarely to cause epidemics in people. And I'm showing just a selection of the many species that are affected by this virus shown here, and using the shaded areas to emphasize what we really recognize. Below is just a fraction of what could be actually occurring. And our tiny window into these early stages for virus emergence is always highly incomplete, with just a massive amount of uncertainty. We have already seen a remarkable range of species affected, however, over the past 2 years, which in our experience never bodes well, because one, the virus becomes increasingly out of reach in terms of control. With the more species and then the more ecological niches that virus is able to fall into. And two, amplification in domesticated animals, especially in intensive production, is another daunting challenge, enabling the virus to spread in new ways that hasn't happened before. And it's important to emphasize that each infection offers the virus an opportunity to change, mutate, adapt, further potentially obtaining new characteristics, including what would be the most worrisome trait, of course, that many have alluded to in the press, is human-to-human transmission. And so while no one can say what happens next, sorry, click next slide, thank you. Human-to-human transmission would, of course, facilitate the type of rapid spread that we saw in the pandemic most recently. So right now we're in a phase in which we need heightened vigilance and very active surveillance in case finding in animals and humans at high-risk interfaces where animals and humans are deeply connected. Next slide, please. So if this scenario is our new normal, we're going to need new tools for surveillance and especially innovative ways to detect outbreaks, including asymptomatic infections that can cause local and regional spread. And wastewater surveillance actually came out of the COVID pandemic as a huge win. This is an excellent tool to track seasonal influenza as well as H5N1. And of course, a concern is that these viruses, these different strains can recombine in an individual that's infected with both at the same time. So that's really useful, but we obviously really need new innovative tools for surveillance that can be scaled to keep pace with outbreaks. And importantly for wildlife, especially help us to measure the impacts of these major mortality events on these species. Next slide, please. And just emphasizing that the impact to wildlife is frightening. We're doing our part at the School of Vet Med alongside the amazing work being done by our labs as outlined by Dr. Hill. We are screening wild marine mammals actively along the coast, even those without any symptoms. We're working with many amazing partners, including the Oiled Wildlife Care Network, that are networked for wildlife rescue and response. And so they're already working together. And we are actively looking for new cases to enable, hopefully, more rapid detection in species that would potentially otherwise be missed. And I think together with a wide array of disciplines across UC Davis and other UC campuses, our new center, NSF Center for Pandemic Insights, is adding a whole new set of tools for H5N1 surveillance and monitoring, as well as other pathogens that we worry about causing pandemics. And I think these new tools can involve pathogen sensors, remote sensing techniques, and even robotics that will be especially helpful in understanding the viruses that are circulating in wildlife. Next slide, please. So I just want to sort of wrap up with, it's really important to face emerging infectious diseases, including the rapidly evolving situation that we have with H5N1 with a very healthy dose of humility, remembering that we only have a small fraction of the understanding that we need to be able to understand the virus and help us consider what could happen next. This knowledge is rapidly evolving and that is natural, and this knowledge is evolving alongside changes in the actual virus and its many hosts. And what we can say for certain already is that we have unprecedented wild mass mortality events in our wild species. And this is a conservation crisis that will have implications for ecosystems and their functioning. And we also obviously have impacts to livestock production, which can potentially threaten food and economic security, not just here, but elsewhere around the world. And I've highlighted some other major unknowns, which will affect our outlook as to what comes next in full. These are just listed as we need to really understand the spectrum of disease and see if there is what we call cryptic or subclinical transmission in some species that could be facilitating spread. We also need to understand the role of immunity and what that means from prior strains and or the current strains. And I think there's major unknowns on that front. We also need to understand the implications of new genotypes as these start to emerge in our dairy cattle industry. And as I said, what about species that are at risk of extinction? What does this virus mean since it has been proven to be so deadly in so many of them? And these long-term ecological consequences are something that we at UC Davis with our partners across UC campuses really need to have an eye on. Next slide, please. I just wanted to also end on some common myths. These are, I think, very common because it's almost impossible to navigate the new information that we all get every single day, even on just this one pathogen. But just clearly H5N1 is a zoonotic disease. It has been so for decades. H5N1 will not burn out on its own. This is a major multi-species pathogen. So there's always going to be a new source of infection with new viruses with their own new mutations and reassortment of these genetic elements that can ensure that immunity is always going to be challenged. So these new versions will ultimately be able to escape immunity that's established no matter how widespread this circulates. And then also H5N1 cannot be stopped with herd immunity. Herd immunity may not even be, alone may not be able to prevent transmission. And avian influenzas are common and are going to be amplified increasingly so in these new settings and these new ecological niches. So thank you. I will now welcome my colleague, Dr. Desai, who will focus on the impacts to humans.
Thanks, Dr. Johnson. I'm Dr. Desai from the UC Davis School of Medicine. And to start, I'd like to give a bit of background on H5 infections and humans in general. So there are actually 9 known subtypes of H5 that have infected humans in the past. And we've seen sporadic H5 infections for several decades, presenting with a spectrum of disease. In terms of H5N1, this was actually first reported in Hong Kong in 1997 in humans. And since 2003, there have been more than 900 human cases reported worldwide. It's thought to have caused the largest number of detected cases of severe disease among the H5 subtype, associated often with high case fatality rates, the caveat being here that this may be inflated due to under surveillance in humans. Before 2024, however, only 1 human H5N1 case had been reported in the US in a poultry worker from Colorado in 2022. While prior strains of H5 have less readily affected mammals due to differences in upper airway respiratories, as my colleagues have already discussed, genomic sequences from virus samples of infected mammals from the current outbreak have revealed mutations in a polymerase gene that may actually explain the ability of the virus to replicate better in mammalian tissues. And so this figure that Dr. Johnson alluded to is from an early report of H5 infections among minks on a farm in Spain, where investigators first noted this activity. Taking a step back, when we think about influenza in humans in general, human influenza strains have been found to use the sialic acid alpha-2,6 receptor in respiratory epithelial cells to adhere to and enter these cells. Sialic acid alpha-2,6 is not only found in the respiratory tract of humans, but also some animals, including pigs, as this figure depicts with the red stars. In contrast, avian influenza uses the sialic acid alpha-2,3 receptor found in birds, pigs, and some specific human tissues, namely lung alveoli, so this would be lower respiratory tract, as well as conjunctiva. And this may explain why humans tend to be infected after exposure to high doses of avian influenza, and in the past have often presented with lower respiratory tract infections when infectants, such as pneumonia. Also of note, with regards to the current outbreak, there has been some evidence from archived bovine tissues that both types of receptors are expressed to a different degree in the mammary gland, respiratory tract, and cerebrum of beef and dairy cattle. And this could potentially be of concern as a future mixing vessel for a novel strain of influenza. But the fact that no sustained human-to-human transmission has been documented in the current outbreak yet may suggest that despite these new mutations that I discussed in the prior slide, the current H5N1 circulating strains have yet to preferentially enter cells using the sialic acid alpha-2,6 receptor. And now in terms of human epidemiology of H5N1 in the US, there have been about 67 cases reported since the onset of this current outbreak. And you can see from this table that most of these cases have occurred in California, with the majority of cases documented among individuals with exposure to dairy herds. Now, at present, the CDC has recommended that healthcare providers consider H5N1 in patients with signs or symptoms of acute respiratory illness or conjunctivitis and relevant exposure history. And so this would include contact with potentially sick or dead birds, livestock, or cattle within the prior 10 days. Now, the incubation period of H5 has historically been estimated at around 2 to 7 days, although this can be variable, anywhere up to 17 days. And in terms of cases that have occurred among humans over the course of this current outbreak in the US, this is one of the first case reports of an adult dairy farm worker from Texas in March of 2024. This was published in the New England Journal, who presented with subconjunctival hemorrhage. The patient reported wearing gloves while working with dairy cattle, but no respiratory or eye protection. Conjunctival and nasopharyngeal swabs from the right eye demonstrated H5 influenza A, with whole genome sequencing revealing H5N1 belonging to clade 2344B. And gene segments were found to be closely related to those detected in Texas dairy cattle and other wild birds sequenced in Texas at the same time. And something of note here was that while the virus identified from the patient did have a mutation associated with enhanced mammalian adaptation, there weren't any changes in the hemagglutinin gene that would affect receptor binding specificity. And so this patient received antivirals and recovered with no sequelae. And just a note here, recall that humans have the sialic acid alpha 2,3 receptor in our conjunctiva, and this may explain why conjunctivitis has been an often reported symptom during this current outbreak. Subsequent reports from August of 2024 documented H5N1 virus in 2 dairy farm workers in Michigan. In this case, dairy worker A presented with conjunctivitis 1 day after a splash on site in the eye while working at a farm that had confirmed H5 in dairy cows. This patient was not wearing any personal protective equipment. A secondary worker B from a separate farm presented with mild cough, shortness of breath, headache, and nasal congestion. This worker did report wearing eye protection and gloves, but no respirator or mask. Now, this slide comes from data that was recently published in the New England Journal at the end of last year, which reports on persons in the US with confirmed H5N1. And so this report actually describes 46 case patients, 20 with reported exposures to infected poultry, and 25 exposed to infected or presumed infected cattle. Among the cases with animal exposures, the median age of patients was 34 years. All had mild illness, none were hospitalized, and there were no fatalities. Most did present with conjunctivitis, about half with fever, and about a third with respiratory symptoms. And interesting, the hemagglutinin gene was successfully sequenced from specimens obtained from about 26 patients, so a little over half. And phylogenetic analysis revealed that all cases had H5 clade 2344B. No additional human cases were identified among 97 close contacts of occupationally exposed workers who were undergoing monitoring or through national influenza surveillance strategies. And this is really consistent with the current sort of lack of evidence for human-to-human, sustained human-to-human transmission of H5N1 viruses in the United States. And in terms of specific occupational exposures, this is a study that was published in MMWR in November of 2024, in which dairy farm workers in Michigan and Colorado that had no reported confirmed illness with H5N1, but did work on dairies with lab-confirmed H5, demonstrated that about 7% of these exposed workers did have serologic evidence of infection with H5 at some point in the past. And among workers who had antibodies to the virus, all reported cleaning within the milking parlor. And none of the workers with antibodies reported the use of PPE that's been recommended, and about half of individuals with antibodies did not report having any preceding illness or symptoms, the caveat here being that their symptoms may have been very mild, and so they may have been missed. So from this data so far, it's clear that there are some occupational exposures that are associated with a risk of infection with H5N1. What's been more concerning more recently are a couple of reports within California of individuals without clear exposure risks. So the first was a child who experienced mild symptoms and was ultimately diagnosed with H5 actually through state and national surveillance programs. There were low levels of viral material detected in the initial specimens collected, and follow-up testing of the child several days later was negative for H5, but was positive for other common respiratory viruses. And so this child recovered from their illness without any sequelae. And, during the subsequent public health investigation, household members who had also reported having similar symptoms did have specimens collected, but all test results from these members of the household were negative for H5 avian influenza. And some family members were positive for the other common respiratory viruses that the child also had. The second child identified later on in 2024 had experienced vomiting and fever after exposure to raw milk. Ultimately, this patient was negative for all flu targets after an investigation by the CDC, but this was felt to not completely rule out the possibility of infection, as it's possible the patient may have had very low levels of viremia. And more recently, the first US H5N1 associated fatality occurred in Louisiana at the beginning of January 2025. So this patient was over the age of 65 and reported to have underlying medical conditions. The patient contracted H5N1 after exposure to a combination of a non-commercial backyard flock and wild birds. And partial viral genome data of the virus that infected this patient indicated that the virus belonged to a genotype related to other viruses recently detected in wild birds and poultry in the US. There was deep sequence analysis of a gene segment from the samples from this patient, which did demonstrate low frequency mixed nucleotides. And this is potentially of concern. And I'm underscoring, potentially here, because these changes may result in increased virus binding to alpha-2 6 cell receptors found in the upper respiratory tract of humans. Though it's important to note that these changes represented a small proportion of the virus population identified in the sample analyzed, and so the changes observed were more likely generated by replication of the virus in the patient with advanced disease rather than something that was transmitted at the time of infection. In terms of diagnostic testing, there have been national flu surveillance and targeted H5 surveillance ongoing. In most commercial or clinical labs, influenza A testing can be done fairly easily, but this doesn't differentiate between what subtype it is. So at my institution, we can subtype to certain influenza A subtypes, though not H5N1. But in California, CDPH requests that if there is concern for H5 or potential high risk exposures in a patient and you have a sample that is positive for influenza A, you can and should send testing to a public health laboratory for further subtyping. In terms of treatment, the mainstay for influenza A in general is always, of course, supportive care. Antiviral treatment is recommended for individuals who are suspected, probable, or confirmed cases. Oseltamivir can be initiated empirically. There haven't been any significant mutations in confirmed cases in the US to suggest that it would be less effective, although there have been cases with mutations to other antivirals to suggest that these may be less effective. And in terms of prevention, my colleagues already did a fantastic job of highlighting important preventative measures in general at the moment. I wanted to show some of the recommended PPE for individuals who are at high risk for exposure, but understandably, some of these may be difficult to implement in certain settings. And there are also guidelines available for post-exposure prophylaxis for contacts of confirmed cases or workers who have been exposed in high-risk settings. And I wanted to just take a moment to highlight this study, particularly in a response to a question that comes up relatively frequently with relation to raw milk. So in this study published in 2024, investigators looked at the persistence of infectious influenza viruses in unpasteurized milk on common milking surfaces in dairy farms, and they found that both H5N1 and H1N1 remained stable in unpasteurized milk and on milking equipment, suggesting that raw unpasteurized milk could be a source of infection or transmission, which is why avoiding raw milk is recommended for this and, of course, a plethora of other infectious disease reasons. And lastly, in terms of other preventative strategies, there are already actually 3 licensed vaccines against H5N1 that exist that were based on early 2000s strains in the US that are part of the National Pre-Pandemic Influenza Vaccine Stockpile. These are not commercially available at this point. There was a study in Nature in 2024 that found that 2 of the vaccines did continue to produce cross-reactive antibodies to the H5 clade 2344B, suggesting that they could potentially still be effective. However, these candidates are egg-based. So in the setting of needing to scale up, this could be challenging. I wanted to note that there are studies underway of mRNA-based platforms that may be useful. Moderna reported encouraging phase 1-2 data that hasn't yet been published, but there are plans for a phase 3 trial that was actually awarded funds at the end of 2024, early 2025 to develop mRNA vaccines for this purpose. So more to be seen on that front. I want to note here that seasonal influenza vaccines that we get are not protective against H5N1, but still very important. And one reason in the context of this outbreak is that it may reduce the chance of human and influenza viruses mixing and preventing reassortants that could be more virulent to humans. And so in summary, a few key takeaways for H5N1 in humans. At this point, there have been mostly sporadic cases among individuals, primarily with known exposures. There haven't been any widespread outbreaks or sustained human-to-human transmission that's yet been documented. And of course, appropriate preventative measures for individuals at high risk for exposure remains critical. Thank you for your interest in these topics, and I'll turn us back over to Dr. McRae.
Thank you very much to all our speakers for sharing your knowledge and expertise. Before and during the webinar, we've been collecting questions and comments, and there are many. There's so much to discuss. I apologize. I will only be able to get to a couple of questions since we only have 5 minutes left. You are welcome to connect directly with our presenters, though, if you want to have a further dialogue. So, one of the questions that came up was, what has the dairy industry's response been to milk testing as a means for standard surveillance? And how is the surveillance financially supported, and is it being done in each of our states?
So I think I can take that one. I can really specifically speak to California. I'm less familiar with the surveillance that's going on in other states, but in California, the dairy industry has generally been very receptive. Can you repeat the question, Deena? Sorry, want to make sure I
Sure.
get all of it.
What has the dairy industry's response been to milk testing as a means for standard surveillance? And how is the surveillance financially supported?
Okay, so financially, the testing is paid for by USDA, so it is not paid, it's not something that the producer gets charged for. And the sample collection at this point, it's kind of a mix depending on where in the processing stream the samples are being collected. So some samples are being collected as part of the routine milk collection that happens either at the tanker truck or at the creamery level. If it's individual milk testing, it's collected sometimes by a regulatory veterinarian, sometimes by a private practitioner, and sometimes by somebody that's on the dairy. But the funding to pay for the testing and for the control measures is not being charged to the producer. That comes from either the federal government or the state government.
Thank you very much, Dr. Hill. This is an easier question, I think. It's more of a yes or no. Does freezing or freeze drying kill the virus? And maybe Dr. Johnson or Dr. Desai, do you want to take that question?
I was just thinking Dr. Stetter was about to.
Oh, okay,
I
Dr.
mean,
Stetter.
yeah, my response would be no, it does not. It is not a proven method for killing the virus. Yeah.
Okay,
Yeah,
thank you
please
very
anyone
much.
chime in if they have new information.
All right, and the last question, if someone could kindly comment on the infection rates outside of the US. We hear so much about what's occurring in our country, but perhaps you could comment on why that's so and any collaboration that's possible with other countries. Dean Stetter might thank you.
I will likely need to call a friend for some more details, but I would say a couple things are obvious that being that the most prevalent species that carry avian influenza are migratory birds that travel all over the globe. We know that this disease exists around the globe. Um, and it really, the question that was asked depends a lot on what species we're talking about. We know that, as we talked about, that certain wild populations, certainly in South America, with both birds and marine mammals, are highly affected with mass mortality events. There are certainly possibilities that it is much more prevalent across the globe that we are not either measuring in our domestic animals or our wildlife. But typically, if there's mass mortality events, that's where veterinarians and ecologists and health professionals would get involved with determining cause of death.
Thank you very much, Dean Stetter. And thank you to all of you again for sharing your knowledge and expertise. And thank you to those who have joined us today. As a reminder, tomorrow you will receive an automated email with instructions on how to provide feedback and claim continuing education units. Thank you very much again for joining, and I hope everyone has a great day.